So I have just started the recording. So the first part we'll be discussing about would be the brain and the neuron. So if you open your Neuroscience at Home packet, you will see there's a smaller bag that's labeled the brain and neuron. If you would just take out that bag, you'll see there are 4 sheets of handouts. Starting with the brain, followed by the neuron, followed by synaptic neurotransmission, and myelination. So we'll be going over these handouts. We'll also be using other materials in the bag, which includes your brain stress ball model, and I believe 2 little pots of Play-Doh. So we'll be using all of these materials. Arjun, are you ready to take it away?
Sounds good.
Perfect. Just let me know when you want to go to the next slide.
Okay, yeah. What we're going to start out with is looking at the different lobes of the brain. And so we're going to use the little stress ball that you guys all have. And so there are 2 different, sections in terms of looking at the different brain structures. And so the one on the left is what is called the cerebral cortex. And so those are basically the outside structures of the brain. Those ones are a lot easier to see on this model because they're more on the outside. And so you can see that the brain is sort of divided up into these different lobes that have been designated or identified, playing a role in certain functions. And so even though we see these kind of outlines that seem to be really concrete, we don't really know the exact outline of these lobes. It's more abstract, but we know that that region itself sort of plays a role in specific functions. And so we're just going to quickly walk through the main functions of these different lobes. So if you look at the front part of the lobe, like right here, we have the frontal lobe, named appropriately. And so that lobe is really important for things like cognition, decision-making, self-control. And so you can kind of think of it as a place where you receive input from the rest of your brain to be sent out to the rest of the body and vice versa. So it's really good at taking a lot of the information from the brain and sending it out and receiving it. And so if we move a little bit behind to sort of this back top area of the brain, we'll go to the parietal lobe. And so that lobe is more specific and it's focused on just the general bodily senses. So, your 5 senses, like touch and taste and smell. The parietal lobe has a role in all of these. And so that would be kind of important, and it's more general than the other lobes, as we'll see. So if you move down a little bit, you can see the occipital lobe. And so that lobe is actually just specific to one sense, and that's vision. So the parietal lobe is involved more so with all 5 in a general sense, but this entire bottom section right here is mainly involved with your vision. And if you move a little bit forward into this sort of middle area of the brain, you can see what's known as the temporal lobe. And so that's really important for your hearing sense. So it's important for, receiving speech and also for forming memories based on what you're hearing. And so, obviously that's a really important part of the brain. And again, it's a bit more specific than the frontal and parietal lobes. And so now we get into sort of these smaller parts of the brain. For example, you have the cerebellum, which is a bit harder to see. It's kind of in this bottom part right here. And so the cerebellum has been mostly known to be important for things like movement and coordination and that type of stuff. But as we're starting to do more research into it, we're finding that it's even involved with more complex things, such as like appetite and like thinking. And so it's kind of a really important part of your brain, despite it being not as big as some of the other structures we've talked about. And then the one more structure I want to talk about from this figure is kind of weird to look at on this model. It doesn't really show up here. So if you look sort of, the camera to focus like right around here, you can see like this little notch inside the brain. And so that is actually around where you have your brainstem. And so your brainstem is actually really, really important because it controls a lot of the basic involuntary functions, such as like your breathing, sleeping, heartbeat. So like it's really important And if you have an injury there, it can be, really life-threatening in many cases. So those are kind of like the basic outlines of the cerebral cortex. And again, those are more, if you want to think of it as like the outside structures. And then what we do in the next figure is look a bit deeper inside the brain. So if you want to think about cutting the brain in half like this and sort of just opening it up, we can see a lot more structures that have, a bit more specific roles, not as big as the lobes, but they certainly are very important for our overall function. And I know the figure has a lot of these parts labeled again, but I think we should only really need to focus on a couple of them. So I think something that's really cool is the hippocampus. You guys can't really see it on this model, but if you want to think it's kind of in this middle here, if you want to think like where the parietal or maybe more the temporal lobe and like inside past that, it's the structure that's really important for memory and emotion and cognition. And so, those are really important instructions for humans, and it kind of makes us, really special and unique from a lot of other animals. So it's a really important structure for us. We have things like the amygdala, which helps out with our emotion. And then something that's also pretty interesting is called the corpus callosum. And so that's the part of the brain that actually connects the two hemispheres of the brain. So you might remember that I said that we don't really have these like sections of the brain when we looked at the cerebral cortex, but we do know that we kind of have a left and right hemisphere of the brain that are somewhat separate from each other. And so the corpus callosum is really important in that it helps to connect these two sides and help to relay information from one side to another, and also, combine information to be sent to the rest of the body.
Sounds good. And Arjun, may I just add two more things? The first thing that Arjun talked about, remember, was the brainstem. So if you look at the bottom of your brain, you see the little notches, right? So the brainstem is actually a very long structure, and it actually extends all to your spinal cord. So if you see the picture on the left, you see, the pons, the medulla, and then later, this all goes along your spinal cord. And now you can see why we call the brain plus the spinal cord to be the central nervous system. Another thing that Arjun talked about would be the hippocampus. I just want to quickly remind you that in our last tutorial, we talked about cognition and memory. And we talked about a patient whose name was HM, right? HM got his bilateral hippocampus removed, and he was not able to form long-term memories anymore. So this really showcases us that, the hippocampus is an integral structure in the formation of memory. That's all. Arjun, carry on.
Can we go to the next slide, please? Yeah, so in the previous two tutorials, you guys went over sort of the basic structure of neurons. So before I sort of get into these different parts, as a reminder, if you want to take a couple of minutes to see if you can label these yourselves. And if you're struggling with a little bit, we do have sort of a term box located below. So I would try first to just get it on its own. But if you need a little bit of reference, I would look below and use those terms if needed.
So you have an empty figure on one of your handouts, and Arjun was talking about under that you have like a word bank. Okay, we'll probably give 1 or 2 more minutes. How's everyone doing with the labeling? Is there like difficult structures? If there are questions or comments, you can either just speak out or you can put them in the chat. We're also monitoring the chat.
I think that should be enough time for everyone to have a chance to try to fill them out. If you weren't able to fill them out, that's fine. We're going to go over them now. So if you could go to the next slide. Yeah. So here are all the correct labelings. So we're just going to quickly go through all of them. You should be familiar somewhat with most of these terms. So just starting from the right and moving to the left, you have the cell body, which is the soma. That's kind of, I would think the most obvious one is just the center of the neuron. It's where all of the intracellular components are, where you have all like the proteins and the structures like ribosomes that are located. And inside of that, you have the nucleus. And so the nucleus is where all of the DNA is stored. It's, the hub of information, and it tells the rest of the body what to do. So it's kind of the most important part, if you want to think of it like that, of the cell body. And so once you extend past the soma, you get to the axon. And so, you might want to think that only this sort of branched out part at the end is the axon, because it's not really shown much, but this entire part, and starting from where the soma ends up to those branches, is actually all axon as well. And so on these axons, we have these large, blank rectangles, and those are actually the myelin sheaths. So we're going to go over what exactly they do and how they work, but basically they help to pass the signals from the neurons a lot faster. And so they basically cover up the majority of the axon, and they are actually made by these cells called Schwann cells, and they're shown by these smaller circles inside of the myelin sheaths. And so those help to produce the sheets to wrap around the axons. And then the parts that we don't see covered by the myelin sheaths, we call those the nodes of Ranvier. And so it's not just this one space that's called the node of Ranvier, but actually all of these interspaced black parts that you can see, we would call those the nodes of Ranvier. And then finally, we have the axon terminals. That is where you would see the signal of the neuron end and be transferred to the next neuron. So you don't need any sort of myelin sheath around there, because you're trying to transport the signal to another neuron. And then, the last structure that we have not talked about is the dendrites. And those would actually be connected to the axon terminal of another neuron. And so they would receive the initial signal and sort of start over this whole process of sending signals through the next neuron. All right, so if you have any questions about that, feel free to put them in the chat. Adrian, I don't know if you want to add anything else.
No. Arjun, a quick question. So if I have a neuron that looks like this, where does the information go in and where does it flow out?
Yeah, that's a good question. So you're going to see information flow in from the dendrites. And so you're going to have the information come through the dendrites. It's going to remain in the cell body for some time. And then you're going to see the signal being sent out again through the axon across these sheets and then out into the axon terminal. And then the axon terminal is going to be connected to another neuron, specifically the dendrites of that neuron. And so you're going to see the axon terminal connected to the dendrites, and that whole flow of information is going to start again. All right, so I think we're all good to go to the next slide. So we're actually just talking about, the axon end being attached to the dendrite. And so that happens through a very specific process called a synapse. And so if you want to look at either of the figures, they're both pretty detailed. You basically have three different parts. So you have the presynaptic neuron, which is the end of the axon. You have the synaptic cleft, which is the area between the axon terminal and the dendrite. And then you have the postsynaptic neuron, which is the start of the dendrite. And so you're going to see information in the form of chemicals and electrical signals from the presynaptic neuron through the cleft and into the postsynaptic neuron. And so the way we do that is through these voltage-gated calcium channels. So I'm sure you guys have at least heard of action potentials. And so when you have an action potential come through the axon with these, different signals, it triggers these calcium-gated channels to open. And so what you see then is that all of these signals that are, kept in these vesicles and moved through the action potential are moved out of the axon through these open gates and are just sort of free floating in the cleft, if you want to think about it. But the distance of the synaptic cleft is incredibly small. So even though they are kind of free floating everywhere, you're going to see, a very good transmission from the presynaptic neuron to the postsynaptic neuron. And so on the postsynaptic neuron, you're not going to just have these sort of open channels, but you're going to actually have these very specific ion receptors. And so what's going to happen is that these signal molecules are going to go from the presynaptic neuron through the cleft and attach to these ion channel receptors. And when they attach, that's going to actually cause the next action potential in the postsynaptic neuron and sort of start over this whole process again. Okay, so on the next slide, we're going to actually try and, build a synapse using a Play-Doh. So if you guys do have the neuron kits, you should have these two little tubs of Play-Doh. One of them has only one color and one of them should have two. So what you want to do is first create the neurons. And so what I would do is I would use that, single tub with only one color to create the two neurons. So you can. I feel like I'm in third grade again, but
I find it really easy, Arjun, to first create like the axon.
yeah, that's true. I don't know. Do third graders even play Play-Doh anymore? I feel like they have iPads. But so you want to have sort of a similar structure to what you're seeing in the figures. Obviously not great, but this is kind of how I made it. I don't know. Yeah. So I kind of have the axon right here, and then I have the postsynaptic neuron over here, and you can see it sort of in this sort of curved shape, and the presynaptic neuron, the axon is more in this like bell shape. And so that's going to be sort of the interaction between those two neurons. And you can sort of see as I have this like to the camera, this C shape, it almost like is like a lock and key fit. And so that little space between those two is where you have the synaptic cleft. All right, so what we're going to do next is try to make a little neurotransmitter. So I would use one of the other colors to try and just make a neurotransmitter, and you can make it however you like. The important part is going to be actually the next step. So I would make the neurotransmitter however big or small as you want, you know, as long as it's not bigger than the neurons themselves. So, you know, like just some little ball like this would be good. So now we have the axon, the neurotransmitter, and we have the postsynaptic neuron, or like the beginning of a dendrite, right? So we said that there's not actually like a direct flow from the neurotransmitter into the postsynaptic cleft, but it actually has to bind to a receptor. So the next thing we want to do is try and make a receptor. And so you want to try to make the receptor in a way that it would be very easy for the neurotransmitter to bind to it. So try to do that for a minute. Okay, so this is kind of how I made my receptor. It's kind of like a weird flower shape, but you can see that when I have the neurotransmitter, how am I going to show this? But there you go. You can kind of see that it fits in here really nicely. And it's like a snug fit. And so that's kind of what you want in general, not just for like a neuron, but for just any sort of signal transduction where you're going from like the extracellular part to the intracellular part. You're going to want a really specific receptor that can sort of catch the signal molecule really easily. You know, so here is kind of what it's going to look like. You're going to have the axon sort of sending a neurotransmitter right here, and you're going to have the neurotransmitter sort of binding to the receptors on the dendrite. So it's usually called sort of a lock and key or a lock and shape, but that's sort of the most important concept you want to understand here is that you're going to have sort of all these different components being formed in ways to where they can interact with each other really easily.
Yeah, and Arjun, may I just add, so we have a little nice figure here that shows what Arjun was talking about, the lock and key model. So if you have a neurotransmitter and it's traveling through the synaptic cleft, right, this little space over here, it wants to bind to some receptors on the membrane of the postsynaptic neuron. And to do that, it's looking for, a really snuggle fit receptor. But why do we want this, what we call specificity of binding? Well, the reason behind that is, a single neuron can be receiving input from a lot other more neurons, right? And these neurons don't have to be sending the same neurotransmitter. So what you want is that, the correct neurotransmitter is binding onto the correct corresponding receptor. So you basically don't end up having some signal confusion that could, really mess up with the activity of the postsynaptic neuron. So you will see concepts like this really all throughout biology. It really extends beyond even neuroscience because, in your body, you have hormones, right? You have insulin, you have sex hormones, vasopressin, and all of these hormones are really traveling throughout your body in your bloodstream. and you want to make sure that they're binding to their correct receptors, so they're exerting their function appropriately. Okay.
Yeah, I think that's a really good point to add.
Perfect. Okay.
So can we move to the next slide, please? All right, so we're going to watch this quick video, and we're going to look at a molecule called serotonin. You guys might have heard of it, or you probably have heard of dopamine. These are molecules that are really important for positive emotions, happiness. So we're going to sort of just look at a brief video about how these serotonin inhibitors work.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss selective serotonin reuptake inhibitors, or SSRIs. SSRIs were developed in the 1970s with the goal of treating depression by increasing serotonin levels. This goal was formulated based on the serotonin hypothesis of depression, which suggests that depression is caused by low levels of the neurotransmitter serotonin. SSRIs work by inhibiting a mechanism called reuptake. In reuptake, a protein called a transporter transports excess neurotransmitter molecules out of the synaptic cleft, usually back into the neuron that released them. SSRIs inhibit the reuptake of serotonin. By inhibiting the removal of serotonin from the synaptic cleft, this causes levels of serotonin in the synaptic cleft to rise. These increases in serotonin levels have been hypothesized to be the mechanism by which SSRIs can treat the symptoms of depression. There are reasons now, however, to believe that there must be more to the mechanism of SSRIs than just changing serotonin levels. For example, when someone begins taking an SSRI, they generally have to wait about 4 weeks before their symptoms improve. Evidence suggests, however, that their serotonin levels rise as quickly as within an hour after taking the medication. Thus, it seems likely that SSRIs may affect serotonin levels, which then leads to other effects that influence the symptoms of depression. Therefore, it's probable that more than serotonin levels must be changed for SSRIs to work, which suggests that depression isn't caused only by low serotonin levels. Additionally, evidence has now emerged that questions the effectiveness of SSRIs. Some research has found they do not work much better than placebos, and may only benefit those who are most severely depressed, which is a minority of patients who actually take the drugs.
Yeah, so that was, I think, a really cool video about synapses, what we talked about, and then also this new type of drug that has been made in the past, I want to say 20, 30 years, and how it was kind of thought to be a really important and powerful drug. But as we've sort of looked deeper into it and its effects and learned more about depression, it seems like it's not as simple as that, right? So we had the example talking about how these neurotransmitters are binding to these receptors in the dendrites, right? But that doesn't always happen. And so equally as important to just cellular function in general as specificity is efficiency. So when you have these neurotransmitters being sent, if you don't have neurotransmitters binding to receptors, you don't just want to waste those neurotransmitters. So what the body does is it actually reuptakes all of the neurotransmitters that don't bind to these receptors, but not everything is perfect. So sometimes you can have these neurotransmitters being, absorbed back into the presynaptic neuron, even if there are receptors available, right? So it's not 100% accurate. And so what the SSRIs try to do is stop that reuptake for a significant amount of time to allow these neurotransmitters to have an opportunity to bind to the receptors available. And he talked about sort of why that may not be as effective in treating depression as they would seem, and also how SSRIs are seeming to not just work through this inhibition of the reuptake, but through a lot of other mechanisms that haven't been identified. So I think that's a really cool topic, and also kind of shows just how complex and how science is kind of changing all the time, and what you think might be effective and really powerful, 1 year might change, in a couple of years, and sort of introduce so many more problems you have to think about.
Welcome to
All right, so now we get to the myelination topic. So this was sort of briefly mentioned when we looked at the biology of the neuron. So myelination is a way to insulate the axon to help preserve the strength of the signal that's passing through the axon. So not only does it sort of insulate it and provide some sort of protection, but it also helps up to speed the process. So if you know about how action potentials work, it's sort of this flow of ions in and out of the neuron to help pass the signal on. And you're going to have to normally do that across the entire neuron. So you can see in the sort of animated picture on the bottom right, you have a D or hypomyelinated neuron. So that would be a neuron with no myelination. And you can see that the signal sort of starts out, but then you have to do just so many action potentials and so many ions have to move back and forth that the signal sort of just, withers out. And it's not as strong as it would be if you had some way to assist it. And so what these myelinations do is sort of decrease the amount of space and the amount of time that you have to have these action potentials happening. And so you can sort of jump across these myelination blocks that are called myelination sheets. So in the left picture, you can sort of see this jumping shown. And this jumping is called salutary conduction. And it's where you have those jumps in those nodes of Ranvier that we just looked at. So instead of having to cross, let's say 100 units of the neuron, if you have these sort of myelination sheets covering up the majority of it, instead of having to do 100 action potentials, you could maybe only do 10. And so that would really speed up the process. And it's even more than just a 10 times difference. It's actually up to about 100 times difference. So you can send signals way faster and they can be just as powerful as they intended to be. And so it's a really great way to help action potentials be sent and to keep the flow of information from one neuron to another.
And a very quick connection here. So as Arjun was talking about, myelination is something that we see in healthy neurons, right? And because it functions to help speed up this the signal transmission. However, there are certain diseases that that involves the destruction of these myelin structures. So for example, if you look at one of our handouts, this is myelination, you'll see there's a disease called multiple sclerosis, in which it's an actual autoimmune condition where the immune system of someone's body is attacking these myelins. And that causes the loss of myelination on the neurons. And you can think that that would result in slower signal transduction. So these patients typically have, blindness, muscle weakness, tremors, and sensation deficits. They're not feeling as much. And this is actually very severe and life-threatening condition. Okay.
Yeah, so one thing that you can also do with the Play-Doh is try to think about how myelination would work. So if you still have your neurons out, or if you want to sort of make them quickly, you can sort of think about and actually see how these neurons can be, covered in these myelin sheets and how that might actually help out with the sending of signals and make it a more efficient process. So. So this is my very poor attempt at making it. Obviously, you don't have something looking like this. It's a lot more evenly spaced and doesn't glitter. So you can see just like in terms of if you want to send the action potential across the blue part, you now have a significantly less amount of blue Play-Doh showing. And so you only have to use sort of that amount of space to send the signal through, which would obviously make it a much faster and a more efficient signal.
Yeah, and here's my model. So what I kind of did was similar to what Arjun has. Like the green parts are the myelin sheath, and then you will want to leave little gaps in between, and those are the nodes of Ranvier. Those are the places where the action potential will be regenerated. So as you can see, you only have a small area on the axon that regenerates the action potential. So the action potential is kind of like jumping from places to places.
Yeah,
Arjun?
so I think that should be the end of this section. So I'm going to pass it off to Adrian to go through the somatosensation part.
Thanks, Arjun. And just a reminder, everyone, so we do have all of these handouts that explain every step that we just did and the biology behind that. So if you happen to have missed some parts, you can always refer to these handouts. Okay, the next step, the next section we're going to look into will be somatosensation. So what really is somatosensation? Well, somato is basically, I believe, Latin or Greek for body, and sensation is our reception of feelings, right? If someone touches you, then you would sense that touch. If you're putting your hand on a hot stove, you'll be feeling the heat. So somatosensation really is about how the brain interprets all of these external stimuli into perceptual signals. So the very aspect of somatosensation we'll be looking at today is how sensitive are our body parts to touch. So if you go into your kit, you'll find another bag that's labeled somatosensation. In that bag, you should have a few worksheets that are labeled somatosensation. You should also have some toothpicks that are bundled together. And then you should have a bar of clay. And you probably will also have a pencil for you to write down your data. So I'll quickly go over what this experiment is really about. So everyone knows that our hands are incredibly sensitive to the things that we're touching, right? So if I'm touching like a coin, like a nickel, if I'm touching it with my fingertips, I can actually feel the patterns on the nickel, right? I can feel the ridges, I can feel the words on the nickel. However, if you slide that nickel from your hand, from your fingertips, to your forearm and to your shoulder, you're feeling less and less detail. So basically, this is telling us that our body, has, different sensitivity at different body parts. Our hands are probably most sensitive, whereas our arms and our shoulders are probably less sensitive to touch. So in this experiment, we're trying to do is to measure the sensitivity of our different body parts. And to do that, we'll build something that we call a 2-point discrimination probe. So what this basically is you take 2 toothpicks and then you stab them into the clay at some distance that creates 2 points, right? So you can see right here, there are 2 points. So basically what this probe would do is you slide on some body part, like let's say my palm, I'm sliding it on my palm, and you close your eyes and you ask yourself, do I feel 1 point or do I feel 2 points? If you're feeling 2 points, that means this body part is sensitive to a separation that's small, right? So you can imagine if your 2-point separation is like very large, let's say several inches, and then let's say this body part feels that you're actually using 2 points, it probably means it's not very sensitive. However, if your separation is very small, let's say it's just a few millimeters, and your body can still tell, well, it's not 1 point, it's 2 points, it means it's very sensitive. So to actually do this, we've prepared these little measuring sheets. And so you can see on each measuring card, you have these little triangles that are separated at different distances, right? So this one is very small, but this one, the 2 points are separated very, very largely. So what I found that's really easy to do is you take out your piece of clay, and then you basically put the clay onto one of these measuring cards. And then what you would do is you would take out your toothpick, so two at a time, and you basically stab your toothpicks into the clay bar at the distances indicated by these cards. So for example, what I have here is something that I've built this morning. So these two toothpicks are, stabbed into the bar of clay at exactly the locations the triangles are indicating, right? So which means these two pieces are separated by a distance of 30. That's a number written down on your measuring card. So which means these two toothpicks are separated at a distance of 3 centimeters or 30 millimeters. So now you will repeat that same process for other measurements, right? So here on your left, you would have, 2 other triangles that are separated at a different distance, and you want to insert 2 new toothpicks so that you can have a 2-point probe that's separated at another distance. You repeat that for all other measurements, which means after you're done that, you should have a piece of clay that serves as a 2-point probe for a variety of distances, right? So these two are separated these two are very close to each other. While these two, this is another 2-point probe that's separated at a larger distance. So I'm going to give you a few moments to do that. Again, the easiest way to do this is just to overlay your bar of clay onto one of these measuring cards. Okay, and feel free to turn on your camera so that we can see your 2-point probe if you want to show them off. And feel free to ask questions about building the 2-point probe. I know this can be a little bit confusing, so we're here to help. And also you can reference this picture that I just took of my probe on the right here. So you will see, the toothpicks are separated at different distances. Another thing that you want to make sure is that for each pair of toothpicks, you want them to be inserted into the clay bar at the same depth, okay? So, I'm talking about, these two should be kind of level to each other so that if you're just touching it with your hand or something, you'll be able to feel, oh, these two, are at the same level. What you don't want to do is if one is inserted more deeply, but the other one is like sticking out, because in that case, all you will be feeling would just be one probe. So you want to make sure that you are feeling 2 probes. Hey, how's everyone doing? How's everyone building their probes? Perfect. I see Arjun has built a nice one. Abby, Simar, any progress? Perfect. All right. Awesome, Simar. Awesome. That looks good. Okay. Perfect. Awesome. Awesome. Awesome job, everyone. Okay. So if you have not finished building that, that's fine. but make sure that you have at least one, okay, so you understand how this experiment is going to proceed. So if you haven't built, all, I think there are 5. If you haven't finished building all 5, that's fine. If you have just one, you can still proceed, but it might be best for you to complete this experiment by yourself. Okay, so what we're going to do now is on your first handout in the somatosensation section, if you look down, there's a data table. So this data table is basically a recording of sensitivity at different body parts to the 2-point probe. So if you look at the first row, it says forehead, right? And then you have a bunch of questions. Do you feel 2 points on your forehead if the probes are separated at 60 millimeters? So that's probably the largest one, right? So if you put this on your forehead, you close your eyes, do you feel 2 points? And for me, the answer is yes, I do. So what I would do is I'll put down yes in this little cell, right? That's under forehead and under 60 millimeters. And then what you want to do is to narrow it down, right? Still using our forehead, you want to use a smaller separation distance. Let's say we use 30. Do I feel 2 points? Well, I do. I feel 30. I feel 2 points at 30. So I'll put down another yes. And then you narrow it down further. You go to 15. And then you will go to 3.75 and then 1.5. So you repeat this process for each of the body parts listed here. You don't have to do all of them, but probably you want to do forehead, you want to do your palm, and you may want to do shoulder or back. Okay, so what you want to do is to write down yes or no to each of these questions. I'm going to give you some time to do that, and I'll also do my little experiment too. So I start with my forehead. Okay, and I'm now doing my back. Okay, we'll give it another 2 minutes. It's okay if you don't finish by then, but probably make sure that you have at least 2 roles filled out. Okay, so how's everyone doing? Does everyone have 2 rows filled out? All right, so I'm gonna carry on. If you have not finished this part, it's fine. You can follow the instructions. Just continue what you were doing after we're done with today's tutorial, but you should be getting the gist out of it. So after we're done recording our raw data, right? So you answered a bunch of yes or no questions to sensitivity at different body parts. What you want to do is for each body parts, let's say, so as you can see from my forehead, I feel at 60, I feel at 30, I feel at 15, but I don't feel at 3.75, and I don't feel at 1.5. So what you want to do is to, for each body part, so for me, for my forehead, I want to circle the smallest separation that I answered yes. So for me, I answered yes at 15, but no at 3.75. So I would circle the one at 15, right? So, and then I carry on. So for my shoulder, well, I don't feel as much. I only feel up to 30, but I don't feel at 15. So I would circle 30. Okay, and you want to do that for each of your body parts. So basically you're circling the smallest separation that you can feel for each body part. Okay, so what we just did was we identified patterns in our raw data, right? So for me, my forehead feels up to 15, but my back only feels up to 30. So what does that mean? Well, the smaller the separation that you can feel means the more sensitivity you have in that body part, right? Because your body is sensitive enough to feel 2 points even at a smaller distance. So for me, this means that my forehead, which feels up to 15, is much more sensitive than my back, which only feels up to 30 separation. So if you go to part 2 of your worksheet, you'll see there's an empty box, and on the right, there's like a weird looking human figure. So what this exercise is about visualizing your data, right? So you've identified the smallest separation you can feel for each body part. Now I want you to transform that into a figure. So draw a little human figure here, but for each area, the more sensitivity you have, the bigger the area. So for example, in my case, my forehead is more sensitive to my back. So I want to draw my forehead to be very large relative to my back. So what you have basically would be a tweaked human figure that really is a sensitivity map. Okay, so how's everyone doing in visualizing their data? Let's move on. So here I drew a very simple figure of my data. So as you can see, my hands are very sensitive, right? I can feel up to a very small distance. However, my back, which is drawn very small in the middle, means it's not very sensitive. So what you have basically is a heat map of sensitivity. And this is what scientists have usually done when they're mapping out the sensitivity of different body parts. So if you look at the slides, this is what we call a homunculus. So a homunculus is a sensitivity map. So in this map, you can see, well, the hands are very sensitive relative to the arms, right? Because the arms are so small. And then The face area of our body is also very sensitive to the 2-point discrimination task. Now, why is that? What is the biology behind that? So imagine if you have a 2-point probe, right? So you have two little toothpicks that are represented by these blue bars. So on our body, the way to feel is to have touch receptors, right? Every touch that we exert onto our body part must be picked up by touch receptors for us to feel it. So if you have very high density of touch receptors, so in the first case here, you see these two receptors are located very closely to each other, meaning that this area is probably very densely packed with receptors. Your 2-point probe are actually getting picked up by two different receptors. And these receptors are going to send different signals to your brain so that you can accurately tell, oh, I'm being touched by 2 points. However, on a different body part where you have less densely packed receptors, such as this one, see the receptors are, are separated very largely, only one part of your 2-point probe is being picked up, which means your body is sensing, oh, it looks like I'm just being touched by 1 point because only one leg of the probe is being picked up. So what we just did, this mapping of sensitivity can be really seen as a mapping of our receptor density, right? Which makes sense. Your hand probably has a lot more touch receptors than the back of your back. Well, the reason behind that is we're always touching and grabbing things and feeling things with our hand, but we don't really do that with our back. Does anyone have questions with this whole experiment and how it relates to the density of receptors? Okay, if you're not done with this experiment, you can carry on after we're finished with the tutorial. But I do want to get to the very last section, which is very interesting, and it's called visual illusions. So we'll be looking at a bunch of illusions and we'll be talking about our feelings. So this is supposed to be very interactive. Feel free to jump in and share. Either you can unmute yourself or you can put in your thoughts in the chat. So the first visual illusion we'll be talking about will be blotted letters. So if you go back into your kit, you will find one sheet that's folded like this, right? It comes folded and there is a seal on the side. And on top it says blotted letters. So the question is very, very easy. Look at this little image on the right and think about, what pattern it is. What is it telling you? What do you see? So just look at this figure, you don't have to stare very hard into it, but just see, what is representing. Does anyone have like a response? Have a guess? Okay. Is everyone looking at this worksheet? Okay, if you have some thoughts, I encourage you to write them down in the white space over here. So for me, what I'm really seeing is it looks like it's a bunch of nonsense, right? It's just a bunch of little pieces that they don't really show a coherent pattern. Does anyone agree with me? Okay, I'm seeing some nods. Okay, so now what we want to do is you want to break the seal on the side, right? So now you want to unfold this sheet. Break the seal, you will see another picture that is exactly the same as the one above, only slightly modified. Okay, so now I want you to ask yourself the question again. What do I see over here? So looking at this figure, what I saw is it now starts to make sense, right? It looks like it's just a bunch of letter B's that are overlaid by some spilled ink. So this phenomenon, do people see this? Do you guys see it's a bunch of letter B's? Okay, awesome. So what this is telling us is actually a phenomenon that's known as the Gestalt psychology. So Gestalt is a German word that means entirety. So it means a group of things. Basically, Gestalt laws tells us in our visual perception, we perceive things as a group rather than individual pieces. And this really explains why when we're just looking at pieces, it didn't make sense. But when we add us in some context, we're supposed to understand this image holistically. There are a lot of Gestalt laws used in daily life. So for example, similarity. We tend to group these things based on how similar they are to each other, right? So here you have a bunch of red dots, but on the bottom you have blue dots. And then the law of pragnance, or the law of good figure, means you tend to perceive things in the shape that you would anticipate. So for example, you don't see this as little segments, but you see as 5 circles, right? These are the Olympic rings. And then you have a bunch of other laws like proximity. Things are closer to each other, are perceived together. So here, if I show you this, most people will say, oh, it looks like 2 groups of dots. Here you have a lot of continuity, right? It looks like they're following some patterns, some trace over here. Law of closure, even though there are gaps, we tend to perceive this thing as a little, enclosure here. So Gestalt laws kind of like are empirical rules as to how we perceive things in daily life. And this can create very strong visual illusions. So for example, what do you see on the image on the left? Okay, does anyone have an answer?
2 faces.
Right. Yeah, I just heard 2 faces, but those are, what's shown in the black figures, right? If you look at the white figure, you see something that looks like a vase. So this is what we call as figure background contrast. And on the right, this is actually my personal favorite. On the right, it looks like to me it's very interesting. So I'm not gonna spoil this, but what do people see on the figure, in the figure on the right?
See a woman.
Okay, you see a woman, right? So to make this easier, this is her chin, right? And she's kind of like looking into the back of the board, right? However, there is a hidden woman here, right? If I remove this cue, so this is no longer the chin, but if you look at this, if this is the mouth, if this is the chin, this is a nose, and these are the eyes, what you're actually seeing is another old woman, right? Do people see that? So, right, if I just tweak the context here, you're seeing 2 images, right, that are actually kind of like embedded into one another. So this is another example of Gestalt psychology. What you perceive is actually dependent on the contextual cues, right? There are a bunch of other visual illusions. So there's the gradient illusion. This bar, it looks like it has a gradient, but it's actually just of one color. So there are a bunch of others. We don't have time to go over into all of them, but I do want to highlight this one as well. This one, if you just look at it, it kind of looks like a duck, right? So here is the beak, here's the head. But if you kind of like tilt your head a little bit, you can also see a rabbit, right? Because these 2 are the ears, and now this is the head of the rabbit. The rabbit is looking in this direction to the right. So this again showcases us how contextual cues kind of alter our perception. There are a bunch of other ones that elicit the Gestalt rules, such as this one, right? There looks like there is a hidden triangle that's kind of like downward, and this is because we're seeing this kind of like illusory contours that are produced by our law of closure. Okay, another thing I want to talk about is called after image. So if you go to this worksheet that looks like an American flag, although differently colored, you will see this is after image. So the way to look at this illusion is I want you to fix your gaze. So really just look at this image and fix your gaze onto the white circle in the middle. And you have to do this, very attentively. So you're not shifting your gaze at all. Just stare at the white circle for about 20 seconds. So I'll be timing you guys. Just look at the white circle in the middle. Keep on staring. Okay, keep on staring. We're very close. The longer you do this, the better the illusion. So keep on staring at the white circle. All right, now look somewhere else. Quickly look into the white space. What did people see? Did you see like a quick flash? Okay, I see some nodding. So what I saw was when I moved my gaze to the white space underneath this image, I saw a normal American flag that's actually red and blue. So this is called an after image effect. I have two more illusions that I want to show you. So just pay attention to the video, and these illusions are very, very strong. So just follow the instructions in the videos.
Welcome to the first in a range of optical illusions by Crazy Videos. Stare at the dot in the centre of the screen.
So
Keep
here's the
watching the
dot.
black and white image of the castle. Keep watching it. It'll soon change to a sort of colour image. You've got to keep watching that dot in the middle of the screen. There comes the colour image. Keep watching that dot in the middle of the screen. Shortly you'll be presented with the black and white image again, but you'll see it in colour. Weird effect, isn't it? It's coming any minute now. Keep watching the dot in the centre of the screen. Do you see the colour image? Glance away and it reverts to black and white.
Okay.
Hope you enjoy.
So that was the illusion, right? Did people see like the green grass, the kind of beige colored walls of the castle? Okay. So what happened is another example of after image, right? Because this figure is actually completely black and white. You know, there's no color. But what we did was once we are kind of like fixating on some color patterns and then it kind of shifts away, we're seeing new colors. So this is a very strong visual illusion effect. Okay, so I want to quickly talk about the biology behind this. So in our eyes, we have the retina, right? That's kind of like lining on the back of our eyeballs. On the retina, there are different photoreceptors. These receptors perceive light information and transduce them into neural signals. So these are the first stage of our perception of color. And on the retina, there are three types of color photoreceptors that we call cones. And these cones, each type is sensitive to a different light range. So for example, the S cone is most sensitive to blue light, the M cone to green light, and the L cone to the red light. Now, so here is here is just a little image of what a cone looks like. You can see there's some little cone that contains pigment over here, right? So if we're just staring at an image, let's say the American flag that looks like this, you see that it's mostly kind of like in blue-green, right? So what we're doing is we're kind of like exhausting our S cones. We're kind of like always using our S cones, and because we're staring for such a long time, what we did was bleaching them. So bleaching is a fancy term for you're kind of like locking the photoreceptors in a in a special phase that kind of like tires them out, so they're no longer functioning as much. Now, if you are completely bleaching your photoreceptors and then you look somewhere else, what's going to happen is you don't have as much S cone activity, right? Because the S cones have been bleached out. So now what's going to happen, you will see the complementary color. So orange, or kind of like in the red range, is the complementary color to blue. If you remove, or if you tire out your photoreceptors that are sensitive to blue, what you will see is actually the complementary orange kind of color. So that's why after we shift our attention, we start to see red colors. And that is basically how the video also works, right? Remember, you start off with a black and white color image, but then you started to see some weird colors. Those colors are supposed to bleach out some of your photoreceptors so that when you shift your attention elsewhere to a new image, these receptors are not working and you're seeing the complementary colors. And don't worry, by bleaching, your photoreceptors are not dead, right? They're constantly kind of like shifting between an active phase and an inactive phase. You're basically just temporarily inactivating them, but they're not dead. Okay, the last one that I want to very quickly talk about is called the floating sausage. So you also have a worksheet on this, on which you have 2 fingers. So this illusion is very simple. What you want to do is you hold your 2 index fingers out, and you are kind of like, hold them several inches in front of your eyes. You want to have your fingers not touch, but you want to leave like a 1-inch distance between them. And now you just look into the distance between the gap of your fingers. If you're doing this right, if you're kind of like moving your finger close to your eyes, what you will see is like there's almost like a little floating piece of finger in the middle of the gap. Are people seeing that? Okay, remember to leave a little gap. Okay. So this is a very simple illusion, but it's actually very strong for me. What's happening here, right? So if you look at this little image here, what's happening is, yes, your fingers are still there, right? There's no little piece of floating finger in the middle, but really this has to do with the working mechanism of our 2 eyes, right? So our 2 eyes are actually perceiving the world in slightly different patterns. And that's because our eyes are not just overlapping, right? We have our nose in the middle. So if you close one eye and if you look at something, let's say if you look at my face or the slides, and then you shift to the other eye, you will see the 2 images kind of like shifted a little bit. They're not exactly the same. So this is what we call binocular rivalry. At any given time, your brain is only perceiving information from one eye. However, it constantly shifts between the 2 eyes. So you are always seeing like a combination of input from your 2 eyes. Though in this example, in this floating sausage example, at 1 second, the brain is processing information from the left. At the other second is perceiving information from the right. And this creates this kind of like overlapping phenomenon that results in this little floating sausage. Binocular rivalry is actually something that is routinely tested in neurology. So here I have included a study that looks into how binocular rivalry, or how this phenomena exhibits in people that is who suffered stroke. So stroke kind of damages a part of your brain. So in this study, they recruited patients who had stroke to the right side of the brain, and they actually found that they don't really exhibit this illusion because their binocular rivalry isn't working as much. There is a decreased, if you read here, there's a decreased alternation rate, right? So their brain is kind of like always looking at the information from one eye relative to the other. Okay, so this kind of wraps up everything that we have for you today. So I want to quickly remind you that there is a makeup tutorial session next week at the same time, 11 to 12:30 p.m. The content is exactly the same, so you can just attend today's. We'll also be posting the recordings to YouTube, and you should be receiving an email from us either this afternoon or tomorrow about the links. Again, I want to remind you that the Brain Bee, if you haven't registered, please remember to register. The Brain Bee will be hosted via Zoom in the afternoon on the 26th. So that's also a Saturday. But that being said, thank you so much for attending today's tutorial. We hope you have fun with building little neurons, learning about neurotransmission. Remember the neurotransmitter that we talked about and the lock and key model. And then we focus on somatosensation, right? I walked you through an experiment where we kind of, really deciphered the differential sensitivity that we have at different body parts. And we related that sensitivity difference to the density of receptors. And lastly, we went over some really fun visual illusions. We looked at, the blotted letters, just our psychology. And then we focused on after image, how bleaching of some of the receptors can lead us to see some other new patterns. And lastly, binocular rivalry using the floating sausage illusion. So thank you so much for joining today. I hope you really had fun. You can hang on to your neuroscience kits. They're just for you. And yeah, are there any questions from the audience? No? Okay, well, thank you so much for joining today. Okay, see, I saw some kind words from Sivani in the chat. Thank you so much. That means a lot.